I'm Daniel Levine, and this is the Bio Report. Cells continuously sense their environment and, in response to stressors, adapt, recover, or die. Soley Therapeutics uses its AI platform to capture thousands of intracellular features and map how cells sense, interpret, and respond to stress. The approach gives Soley the ability to pursue previously undruggable targets. It's generated more than 10 novel oncology programs in less than 2 years and advanced 2 first-in-class experimental small molecule therapies toward the clinic. Yerem Yeghiazarians, co-founder and CEO of Soley, discusses the science behind the company's first-in-class lead candidate, the applicability of Soley's platform to a broad set of diseases, and the capital efficiency of the company's approach to drug development. Yerem, thanks for joining us.
Thank you for having me today.
We're going to talk about stress biology, Soley, and your efforts to develop novel therapies using a platform that aims to overcome the limitations of single-target approaches. To start, let's talk about cellular sensing. What do you mean by that?
So it turns out, Danny, that cell stress is central to many different diseases. Cells continuously read their environment and shift towards adaptation, recovery, or death. What we're doing at Soley is that we use that built-in cell-sensing capacity as the starting point to discover drugs. And the goal is to measure how cells respond to drugs in their environment, not just whether a molecule binds a target or not. So what we do, if our intent is to discover cancer drugs, is we find drugs that selectively increase the stress level in cancer cells to kill it. And in non-cancer indications, non-oncology indications, we find drugs to decrease the stress level in a cell to make that cell more functional, survive longer, and work better.
How did you come to see this as a basis for an entire drug discovery company?
So my background is in cardiovascular medicine and stem cells. For the last 2 decades, I've been the founder and director of the Translational Cardiac Stem Cell Program at UCSF. And my co-founder's background is very different. His background as a cancer molecular biologist. So we came at this from different angles: cardiovascular medicine, stem cells, and oncology. And we discovered that across many of these diseases and others, cell fate is shaped by how cells interpret stress, not by any single pathway. And this was the moment that it suggested to us that the platform could be much more powerful than just one disease entity, but it could be used as a platform for wider drug discovery, both in cancer and non-oncology indications.
You're using AI and imaging to capture and analyze thousands of intracellular events in live cells. What are you actually measuring?
So when a cell is exposed to a drug, it senses the drug in the environment and information starts to flow within the cell. And the changes in cell state over time, including stress, adaptation, and the trajectory towards whether or not the cell is going to survive or die is what we measure. We measure thousands of image-based features tied to protein expression, localization, transport, aggregation, and other stress biology. And we use multimodal imaging. Sometimes we use live imaging with no stains, and sometimes we use autofluorescence to stain specific proteins that move around a cell that dictate cellular fate. And importantly, with our live imaging technology, we can do this over different doses and different time periods, not just at one static time point, which gives us a lot of additional information.
Well, walk me through the process. What does this look like experimentally?
So we have many different types of cells at Soley Therapeutics, and we now have close to 500,000 small molecules and growing. So we have, using our first-in-class wet lab and automation systems that we've put in place, we can screen up to over 100,000 drugs on a weekly basis. So we essentially expose the cells to compounds across doses and time points. We do all this in our lab, and we image them using our multimodal imaging, as I mentioned, including live imaging and stain images. And these are what we use as the data sets to train all of our foundational models for our computer vision AI ML. And we just had a recent paper a couple of months ago that we published on this topic.
So there are many different forms of cellular stress. This could be hypoxia, metabolic stress, DNA damage, or inflammatory stress. Do you look at these one at a time? Do you look at these simultaneously? And how do you decide which are most relevant to a given disease?
That's a good question. So our work at Soley over 16 years ago, even though Danny, the company is only 5 years old, we've been working together with my co-founder on this for over 16 years. So our initial work began with oxygen and nutrient as the source of stressors. And in fact, we started this work in 2010, and 9 years later, a Nobel Prize was awarded to those who discovered how cells sense oxygen in the environment. 2 years later, another Nobel Prize was awarded to scientists who discovered how cells sense temperature, touch, and pressure in the environment. What we've been doing essentially for the last 16 plus years is to figure out how cells respond to drugs in their environment. So even though we started with oxygen and nutrient sensing, we have now extended our technology to how cells sense drugs in their environment. And it turns out that every drug, when a cell senses exposure to that drug, creates a unique map, which we call the Soley map, of information flow through the cell, which we use for our drug discovery purposes.
So your process really flips drug discovery on its head. In typical drug discovery, you think of people screening thousands of compounds, even millions of compounds through a high throughput screening against a specific target and look for what's a hit. Your model is a drug first, disease later approach. Once you find a phenotype shifting compound, how do you systematically work back to mechanism of action and target?
That's a great way of explaining technology. So the way I've been trained for 30 years as a practicing clinician and scientist, typically the way drug discovery happens, as you know, and as you alluded to, we think of a disease to study, and then we think we know a target within that disease, and then we try to find a drug that binds that target, and then we optimize that with AI ML these days. There are a lot of technologies, as you know, and then once we have a drug that binds a given target for a given disease of interest, then we start doing the downstream experiments. That works really well for many simple monogenic diseases, and as a clinician, I'm the beneficiary of using that daily in my patients, such as cholesterol lowering. However, in complex diseases, whether or not it's cancer or other neurodegenerative diseases or other complex diseases, despite great advances in technology, we're still not 100% sure what is the target to go after in these indications. So my co-founder and I decided to flip the drug discovery paradigm 180 degrees. Instead of starting with disease and target and drug, let's find drugs that give rise to these unique Soley phenotypes. And then we put in place completely novel processes to figure out how that drug works in a cellular level, not at a target level. And we're able to do that. And that has been one of the Achilles heels of phenotypic drug discovery. But at Soley, we're able to successfully overcome that by the target deconvolution processes that we put in place. Notice I still haven't told you which disease we're going to select. Once we have a drug that is druggable, that is well tolerated in preclinical studies, that we know how it's working at a global cellular level, we've put a completely novel bioinformatics process in place to then decide which disease and which patients with that disease to treat in our upcoming clinical trials. So you're absolutely right. We have flipped drug discovery 180 degrees, and we're excited with where we are.
Given that, what does it take to make a decision about when to advance a molecule into development?
Well, I think it's first led by science and the data. And we look at the biology, we look at the preclinical data, but we also look at unmet needs and practical clinical paths forward. So we combine our in-depth scientific know-how, our scientific foundation, and then we see what the data is, what the data looks like, what's the unmet need, and then we make both a scientific as well as a business decision to bring some of these assets forward.
Is there a good example of a compound where the self-stress signal revealed something surprising about mechanism that you wouldn't have seen with a target first approach?
Yes, absolutely. In fact, we one of the first assets that we're publicly speaking about is in cancer. It's an oncology cancer drug that is actually a small oral small molecule that is targeting a previously undruggable target. We're actually presenting this in the next couple of weeks at AACR in San Diego on April 20th. And what this small molecule does, it modulates CCAP2, which is, as I said, a previously undruggable cancer drug. So the platform is able to not just discover novel drugs, but also lead to novel biology that allows us to discover new drugs against undruggable targets, as we're showing in our AACR poster coming up.
And does the platform ever lead you to find molecules that would sit outside what would be considered the known druggable space? And how do you think about developability in those cases?
Yeah, absolutely. So the drug that I mentioned we're going to publicly talk about at AACR is doing indeed that. And the platform is powerful enough for us to be able to find drugs against undruggable target, and that is part of the value proposition. Finding actionable biology beyond conventional target space.
Your lead experimental therapies are both small molecule drugs targeting cancer. The first, SOL001, is a program in the aggressive blood and bone marrow cancer known as acute myeloid leukemia. Biologically, what is your compound modulating in AML, and how did the platform point you there?
Great question. So the AML program emerged from Soley's stress induction strategy. And it was selectively pushing already stressed cancer cells past the threshold and killing them, which is what's desired. The drug is also applicable to not just AML, but other liquid and solid tumors. We decided, again, from a science and business strategy to select acute myeloid leukemia, which is a rare orphan indication. And the preclinical data looks outstanding for us to select that as a disease entity to bring it forward.
And what's known about the safety and efficacy of SOL001 from studies you've done to date so far?
We have seen a strong preclinical anti-tumor activity and non-cytotoxicity in normal human cells, as well as good tolerability in non-tumor-bearing mice, and also, importantly, synergism with standard of care in acute myeloid leukemia drugs.
Now, what's the development path forward?
So we're currently in IND-enabling studies, and we plan to submit an IND application by the end of the year, followed by initiation of our phase 1 clinical trial, if cleared. The current plan is to move into the clinic in AML first with this drug, and in the future, hopefully, once the drug is approved, we'll see how the clinical data is, of course, then one can think about line extension, and we essentially have a pretty good idea which other liquid and solid tumors this drug might actually be applicable to.
You've got a second program, SOL002. This is in development to treat solid tumors. What's the difference between 001 and 002?
Yeah, so good question. So first, Danny, over the last just 2 to 3 years, with a small team, with a small relative budget in drug discovery space, we have over 10 novel cancer drugs discovered already. With the funding that we have selected to bring 2 assets forward to clinic. So this one that you're alluding to, SOL002, is intended to also be first in class, very distinct in mechanism of action compared to the first drug that we just talked about. And we have decided to bring this one against a solid tumor. And this is a completely different scaffold, completely different drug, completely different patent, completely different MOA, and it is also first in class against a completely different indication. So my expectation is that by 2027, we will have two concurrent trials with two different drugs, two different MOAs, all discovered at Soley. We do not in-license any of our drugs. And these are the two that we're bringing forward. And in addition to these two development candidates, Danny, as I said, we literally have 10 more on the way.
002 sounds like it might be broadly applicable. How do you determine which indications you'll pursue first?
So we have tested many different cell lines, and we look at many different things, including genomic background of responsive versus unresponsive cell lines. And so we have a lot of proprietary technology that allows us to figure out which diseases to go after. Right now, for SOL002, we're looking at head and neck squamous cell cancer as the most likely initial indication, which is, of course, a solid tumor. So we'll have one trial in liquid tumor, the first one in AML, and the second one in a solid tumor indication.
You've publicly mentioned that you're looking at everything from neurodegenerative and metabolic indications, including obesity and hair loss. What common stress biology threads link such diverse groups of diseases?
Yeah, that's also a great question. Remember, we started by saying cell stress is central to so many diseases. So in cancer, we're selectively, and the key word is selective, we're selectively increasing the stress level in the cancer cells to kill it because you don't want to have the atomic bomb. You don't want to have a bleach, right? You want to have selectivity, frankly, within tumors, but also against non-tumor cell lines to have a therapeutic window, which is exactly what we have with our drugs. In the non-oncology space, cell stress is central to how so many of these other diseases develop. For example, Parkinson's disease. The reasons why patients develop Parkinson's disease is that the cells in the brain that produce dopamine over time are exposed to stressors, and over time they become more and more and more stressed and dysfunctional, so they stop producing dopamine, and the patients start to have Parkinsonism symptoms. In patients with hair loss, the hair follicular cells become more and more and more stressed out to the point that they stop producing hair. In patients with diabetes, what happens in their pancreatic islet cells is that those cells, for many different stressors over time, become more and more and more stressed and they stop producing insulin, and so on and so on. So this is commonality of diseases. And that's essentially the common thread across many of these non-oncology indications. And given the funding that we have selected neurodegeneration, with Parkinson's being the top priority at this time, but it's also applicable, frankly, to many, many other neurodegenerative diseases. Our platform is, including Alzheimer's disease, a very differentiated approach than what most people are doing with the beta amyloid story, Huntington's disease, muscular dystrophies, Lou Gehrig's, ALS, you name it, we can study it. But we're staying focused on Parkinson's. We have a completely differentiated platform for obesity, which is very different than all the GLP-1 and GIP activity ongoing with many other companies. And we already have some hits at this stage that appear to be as good, if not better, frankly, at this preclinical stage, than even some of the oral GLP-1 agonists that were recently approved. In hair regeneration, we already have some hits that appear to be functioning better than the two FDA-approved drugs in this space for hair growth and regeneration. So the platform is based on a scientific foundation, Danny, and the scientific foundation is applicable to a broad range of diseases, whether or not it's cancer or non-cancer. And within that, there are many, many other indications that the platform can be applied to.
And are there early non-oncology data you view as platform validators, even if they're still pre-IND?
Yes, absolutely. Great question. We have actually published many of these in peer-reviewed journals in two indications. One in a heart attack setting, in cardiovascular setting, and another one is stroke setting. So all the in vitro to in vivo validation has been established, validated, and published in peer-reviewed journals in both cardiovascular and stroke data. And we're excited to then apply that same scientific foundation to some of these other diseases, including Parkinson's, obesity, metabolic diseases, and hair regeneration. So yes, we've published many of these in peer-reviewed journals over the years.
And is there anything known about the specificity of these therapies? Is that something you look at, or do you find that you risk off-target effects with them?
No, we actually have a lot of granular information about how the drug actually works at a global cellular level rather than a target. In fact, you can take, Danny, any small molecule, any drug that's been approved over the many decades, and it turns out even if a drug binds an upstream target within the cell, it does 30, 40 other things. So our platform can delineate that from day one. We can tell you this is how the drug is working at a global cellular level rather than focusing on just one target, because as we all know, every small molecule, in addition to binding an upstream target, binds 30, 40 other proteins within a cell. So instead of simplifying and reducing the MOA, the mechanism of action, to just one upstream target, we actually tell you how the drug really works at a global cellular level. And in fact, we've taken drugs off the shelf, FDA approved, and we also showed that in addition to what we believe the intended target or the MOA is, these drugs do many, many other things. And it turns out all the downstream effects, the flow of information that I alluded to earlier, we're correlating that to clinical outcomes for efficacy and toxicity. So this will become a very powerful data warehouse that one day it will have the power to actually early on predict what may actually become a druggable compound versus a toxin. So we don't spend as an industry decades and billions of dollars wasting money on something that has a 95% failure rate at the end of the day.
How are you thinking about partnering? What kinds of partners and what indications are you most interested in seeking partners for?
So we have a lot of partnership opportunities, as you've alluded to. We have the oncology program, we have the non-oncology program. Within non-oncology, we have different types of programs. We have assets that are headed to clinic. We have assets that are preclinical and the IND stage, and we also have this powerful platform. So we're open to all these conversations with pharma. Because we have some strengths, but they have many other strengths that we just don't have here at Soley. So I think, given this differentiated orthogonal platform that is working amazingly well to date, we're excited to, both from oncology and non-oncology perspective, see what the opportunities would be for early partnerships around differentiated preclinical programs. For the oncology assets, we have the funding to take two of our development candidates to clinic, generate human data, and I think that will interest pharma even more, frankly, at that point from an oncology perspective as well.
Given the broad applicability of the platform, are there programs that you're focusing on for your own development versus partnering?
At this time, we plan to keep our programs that are core to providing providing support for the platform, building enterprise value at Soley. But as you stated, we have many, many partnership opportunities where working with outside partners would have value add to bringing additional assets to clinic, treating additional patients, doing additional clinical trials, both in oncology and non-oncology. So decision is strategic, not just scientific, but we're happy to discuss this with any and all pharma companies to see what will work for them and what will work for us. And most importantly, what can we do to accelerate drug discovery to help our patients?
So a lot of the early AI platforms used for drug discovery have been pitched as faster, cheaper ways of getting into the clinic. How does your platform compare in terms of cost and time to traditional drug discovery?
That's a good question. So first, I want to say that we are not an AI first company. If anything, we're a science first company. It's the scientific foundation of literally collectively over 40, 50 years of research between myself and my co-founder and 16 years of working on this together. It's the science, Danny, that's driving all of this. AI, ML, and automation is used to accelerate and scale up everything that we're doing. So our story is not just speed for speed's sake. It's better biological starting points. In fact, over 16 years, Danny, the scientific foundation that started in my lab at UCSF has not deviated one bit. It's just that over time of more funding and with automation and AI ML, we have accelerated all this to the point that we can now screen 100 plus thousand compounds a week and accelerate the work that we're doing. But I want to make sure that everybody understands that this is a science-first company with decades of scientific insights, in-depth scientific insights into how cells can be used as the strongest biosensors to teach us about how we can discover drugs in an orthogonal way.
But can anything be said about its cost effectiveness?
Yes, it's very cost effective. With a staff of less than 40 people, Danny, as you know, we've raised $290 million since the founding of the company, including a $200 million Series C recently. We have over 10 new cancer drugs in less than 2 years. And, we have significant amount of capital left to actually allow us to complete 2 clinical trials in 2 different oncology programs. This is unprecedented. It is speedy, but more importantly, and hopefully, once we'll see the clinical trials, it'd be effective. Once we show clinical trial data, then it's the speed, effectiveness, and the efficiency by which we can discover and bring drugs forward. And remember, none of these assets that we're bringing forward, Danny, have been discovered elsewhere and in-licensed here. We discover and develop and bring everything forward ourselves from scratch.
You've been able to raise significant capital in what's been a difficult environment for biotech financings, particularly for early stage companies with people who haven't had a track record of commercial success. Why do you think you've been successful with the investors?
I think it's the differentiated scientific thesis. It's a novel approach, orthogonal approach, and we're showing the data. It's not just an AI story. We're showing data that we can actually do this. We have drugs that are in IND enablement. We're heading to patients by the end of this calendar year. So I think when investors look at us and look at our platform, they see decades of scientific know-how. They see a platform that is now automated. They see a platform that's actually producing data. And when you look at the data, they're super impressed because the data, as I said, looks very promising. And we're very excited to actually, the next step is to get to patients. Once we get to patients and show clinical data that's positive, hopefully, the trial has to be done, then I think, everybody who says, oh, it's difficult to do drug discovery in a different way, instead of disease, target, drug, we've flipped it 180 degrees, I think they'll see the light that it is possible to do drug discovery in a completely orthogonal, differentiated way. So we're very excited about it.
What advice would you give other founders building deep science platform-centric companies on how to raise money today?
So the key, I would say, it took us over 10 years between my co-founder, Kurosh Ameri, and I to actually discover and develop this platform and validate it at UCSF. Frankly, I raised about $7-8 million of philanthropy money to validate the science and the platform before we were so convinced that it would be applicable to novel drug discovery. So first, lead with real biology and real execution, and not just abstract platform claims. Because, that may sound good at the beginning, but then if you're not producing any data, then you're all going to look bad. So make sure your science is solid. If you believe your science, then execute. Create a platform or create what you believe is going to be a game changer, and show that the platform can actually produce assets, not just data for data's sake, but assets that are that can be brought forward to clinic. Be disciplined about your story. What's new about it, what's proven about it, and what milestones come next, and be very strategic about how you do everything that you think, plan it ahead, 6 months, 12 months, 3 years, 5 years, and stick to it. It's not easy, but so far so good for us, and I'm actually really excited to get to clinic and start treating patients. We'll see how it goes.
Yerem Yeghiazarian, co-founder and chief scientific officer of Soley Therapeutics. Yerem, thanks so much for your time today.
Thank you so much, Danny. I really enjoyed the conversation.
Thanks for listening. The Bio Report is a production of the Levine Media Group. To automatically download this podcast each week, subscribe to our RSS feed or through iTunes or other podcast manager. To join our mailing list, go to levinemediagroup.com. We'd love to hear from you. If you want to drop us a line or are interested in sponsoring this podcast, send an email to danny@levinemediagroup.com. Special thanks to Jonah Levine, who composed our theme music, and the Jonah Levine Collective, which performs it.